Today's video report of Group 1 cram staining. Here is some laboratory tools and chemicals. First, we need to prepare the smear. Of 3 samples, we start with pickle sample. Using a metal inoculation loop, sterilize it on the flame of alcohol burner until it turns red. After allowing inoculation loop to cool, use it to transfer pickle sample. Fresh sample must be used. Aged cells lost their ability to retain stain. The bacteria then fix to the slide by quickly passing the slide through the flame few times until it totally dry. When finished, flame the metal loop to sterilize. The next sample is rodent meat sample. We do same as with the pickle sample. Also sterilize the inoculation loop on the flame of the alcohol burner until it turns red. After allowing inoculation loop to cool, use it to transfer the rotten meat sample. When finished, flame the metal loop to sterilize again. Then fix the bacteria to the slide by quickly passing the slide through the flame few times until it totally dry. The last sample of cram staining is coliform sample. We do the same thing as to previous sample. But in this time we need to pass the open top of the glass tube through the flame to kill any microorganism that may contaminating the open of the tube and could potentially contaminate the sample. Use the inoculation loop to transfer the sample to the slide. When finished, sterilize the inoculation loop on the flame again. Then also fix the bacteria to the slide by quickly passing the slide through the flame few times until it totally dry. We continue with the procedure. We add few drops of crystal violet stain to the bacteria. Let it stand for 2 minutes. Then we rinse the slide under the open faucet dripping. Let the slide at a 45-degree angle until most of the stain has been washed up. Next, add a few drops of creme iodine to cover the bacteria and let it stand for 2 minutes. Iodine rose at unmodern to combine with crystal violet to form diiodine insoluble complex. Rinse the slide under open faucet dripping. At this time, the cells still appear purple. Now add few drops of decolorizer, 95% ethanol. Let the slide at a 45-degree angle until the flow is clear, usually 10 seconds. Gram-positive bacteria still retain the purple of the crystal violet, whereas gram-negative bacteria do not. This is the most important step because if it over-decolorizes, some gram-positive bacteria will lose the purple and become gram-negative bacteria at the second stain time. Counter stain by adding basic fusion stain. Adding 2 drops and then remove it to avoid water mixing in this. And then add basic fusion again and let it stand for 2 minutes. After 2 minutes, completely rinse the slide under the open faucet dripping. Gently blot dry the slide by paper. Use the microscope oil to the slide, and then use the objective lens 100x. Gram staining used to identify that bacteria is gram-positive with the purple of crystal violet or the gram-negative with the pink of basic fusion. Now is the result of pico sample first. In pico sample, we have both positive bacteria and negative bacteria, but positive bacteria greater than negative bacteria, so pico sample is gram-positive bacteria. Next is the result of rotten meat sample. We both have positive and negative bacteria in rotten sample, but negative bacteria greater than positive bacteria, so rotten meat sample have gram-negative bacteria. Final is the result of coliform sample. This coliform sample quite blurred and small, but we also can see that we just only have the pink color in the sample. So coliform sample have the gram-negative bacteria.